today we're going to talk about my two cats, latency and persistence. It's okay. Their real names are Ava and Jaja, for those of you who know about the Gabor sisters. This might be a question, but it'll depend on Dr. Racaniello. He sort of said, how are you going to work this in? And I said, I'll find a way. 1962, Neil Sedakis recorded a song, which was breaking up is hard to do. And that's really the essence of what we're going to talk about today. We're going to talk about what happens and what are some of the effects of virus long-term infections, persistent infections, where virus particles are made to some extent, latent infections, where sometimes virtually nothing is made, sometimes a few proteins. But perhaps the most amazing part of latent infections is the fact that, for the most part, the virus finds a place in the host where it can lay dormant for the life of that animal. In our case, it's man. So breaking up, that is, clearing the virus, is an extraordinarily difficult event in some cases. Never happens in most cases. And even when it does happen, we don't know that we've cleared the infection, and that pertains to the hepatitis viruses. Okay, so we heard a little bit about acute infections and persistent infections from Dr. Racaniello last week. And to make life simple, it doesn't hurt to go over it again. And I want to remind you that an acute infection is a natural infection that is usually rapid and self-limiting. You get hit, you get sick, you're better. Persistent infections, on the other hand, are a natural infection that can be long-term in nature. So it's slow. Viruses tend to replicate over a long period of time. Sometimes they're abortive. The virus doesn't complete its replication cycle. Sometimes the virus associates with a host and something happens and its genetic information is disrupted. And sometimes they're transforming events. That is, they cause cells to become malignant. You saw this last week as well, and I want to go over it just briefly and remind you that the acute infections are short in duration. Virus replication and symptoms occur. Replication occurs before symptomology, almost always in all cases. We have some instances of long-term persistent infections, such as lymphocytic choriomeningitis virus. And it just sort of smolders and it stays there and it does really bad things to you. And over a period of time, if you're a mouse, you die. And then you have herpes simplex virus, which is present in really all of you, whether you like it or not. And that's a persistent infection that lays latent and dormant in your neurons, in your ganglia, in both your in both your sympathetic and sensory neurons. And the hallmark of herpes simplex virus, and CytoMegalo for that matter too, is what we call recrudescent infections. And that's waves, periodic waves, where the virus is reactivated and sheds, and you have a noticeable manifestation, a cold sore, for instance. In the case of chickenpox virus, varicella, it's shingles. In the case of CMV, it's usually silent, but the virus is highly infectious. In the case of varicella zoster virus, we don't see any inapparent shedding like we do with herpes simplex virus. So there are people who are you're all carriers, pardon me, but there are people who have herpes simplex virus and transmit it without signs of overt lesions, and those are shedders. So we see that wave. And then there are these persistent infections that are very, very slow, such as the measles association with SSPE, and we'll go briefly through that again today. HIV infection, and Dr. Racaniello will address that in greater detail later, and the human T lymphotropic virus, which is a relative of the HIV. Okay, with viruses such as rhino, influenza, and even HIV, there are small changes that occur because these are RNA genomes, and the replication machinery is not perfect. And the resulting viruses are selected, and they're selected for small changes in antigenic variation. and they survive. And that leads to shedding of virions that are resistant to clearing, because whatever antibodies are made are not effective. So in some cases, infections can linger on longer. And that's antigenic drift, those small changes, and selection process that allows for the viruses to escape. Persistent infections occur when primary infection is not cleared by the adaptive immune response. So everything that we've learned about that's so good and that works so well doesn't always work so well. And depending upon when the immune system is activated and how vigorous that response is, an infection may or may not be cleared. And what happens then is that virus genomes, that shouldn't be, it's virus genomes and or proteins, continue to be produced for years. There are instances of viruses that are persistent, such as hepatitis B and hepatitis C, where viruses are produced for years. So the liver is infected, and virus is constantly produced, and yet the virus remains, and it remains immune to the host. Now, what's the difference between a chronic and a latent infection? Chronic infections, which are persistent infections, are eventually cleared. So eventually something happens, the host develops some sort of reasonable immune response and removes the infected cells from the population of whatever organ that might be. However, in the case of latency, that's not what happens. In the case of latency, those virus genomes persist for the lifetime of the individual. And where they persist, in what organ, what the target tissue is, varies for various viruses. What are some of the general properties of latent infections? Well, gene products promoting replication are generally not made. And that's a generalization. And of course, like everything else in biology, it's not an absolute. Everything that we say is an absolute turns out not to be. Gene products that are made are found in very low concentration. And when they are found, they're frequently in the wrong place. So instead of being where the virus can use those proteins either to replicate or to transcribe or to transmit some aspect of its protein repertoire, it's just not there. Cells with latent genomes are masked from immune surveillance. They're hidden. And viral genomes persist intact to reactivate and spread to a new host. So the virus is not there to die. It's not there to go into that particular tissue, that particular cell type, to never come out. It really wants to reactivate. And the only exception that we know about is measles and SSPE, where the entire measles genome is not present, only parts of it survive, and they encode these proteins that are passed from cell to cell and cause SSPE. Okay, so what are some examples of latent infections? One that we'll talk about is the Epstein-Barr virus. It's the causative agent of mononucleosis. It's the causative agent of nasopharyngeal carcinoma. It's the causative agent of a proliferating B-cell lymphoma and Hodgkin's disease. And that's enough. All right. And when the virus is latent, it has novel transcription and replication patterns. And generally, it makes no new virus. But it has 3 different forms of latency. And we'll talk about those in a little more detail later on. And different transcription profiles to match those 3 different forms. What is interesting is that the genome replicates. And the reason that the genome has to replicate is that it can't be partitioned to another cell. That is, if the cell divides, it can't go with it if it doesn't replicate. Otherwise, it gets diluted out. Think about that. We'll go through that example a couple of times today. If you only remain in the cell that you infect and the cells divide, eventually there are more daughter cells than there are progenitors. Adenovirus is most frequently associated with terrible upper respiratory illnesses, can be isolated from lymphoid tissue, the adenoids in the back of your throat, and tonsils. So the virus is there at very, very low levels and can persist latently. And when you take those tissues out, you find that most of the virus is actually present in lymphocytes in those organs. And when you culture the organs in tissue culture, you can't get virus out. They don't produce virus. So that tells you something about the milieu in which the virus sits and the cells in which it sits, and how there must be some contributory factors, some relationship between the virus and where it is in the host that allows it to replicate at a very, very low level and persist. Here's an extensive table from your book, and it points out a bunch of interesting things. And it tells you that, for example, hepatitis B can be found in the liver and lymphocytes, and the consequences of it being there a long time are cirrhosis, a fulminating infection of the liver, and later hepatocellular carcinoma. And we believe that one leads to the other as a result of inflammation. So you see the tie-ins to all the things that Vince and I have been talking about over the last couple of weeks, and how viruses elicit immune responses, and sometimes what the host does really screws things up. Herpes simplex viruses are found in sensory and autonomic ganglia, and when they erupt, they form cold sores or genital herpes. Papillomaviruses are found in epithelial cells, and they're found predominantly in the basal layer of the replicating epithelium. And the sequela of long-term infection is papillomas, not caused by frogs, but by virus, and carcinomas. And that depends on the type of virus that you're infected with. So there are over 100 different types, and we'll get onto that in a little while. And then more recently, it's become very apparent to us that there are a family of human polyomaviruses, and two of them are on this chart, and I'll talk about another one that was found 4 years ago, which is, yeah, I think it's more interesting than these two. But they lay in the kidneys in the central nervous system, at least JC does. And they're, for the most part, dormant. But it's been noted that people who are immunosuppressed for one reason or another, either because they're infected with AIDS virus, or because they're on an immunosuppressive regime, because they're a transplant recipient, frequently develop a demyelinating disease called progressive multifocal leukoencephalopathy. So you'll each be asked to pronounce that at the final on your way out. Or hemorrhagic cystitis. And these only occur under conditions, for the most part, of some sort of immune suppression. So think back about what Vince said the other day about measles and immune suppression. There can be some other force in nature which throws everything out of balance. I've told you about herpes simplex, and let me just move on to HCMV. And you'll see, look at the just sheer number of cell types in which this virus can persist. Kidneys, salivary glands, lymphocytes, macrophages, stem cells, stromal cells. It's found in breast milk. It's found in semen. It really gets around, all right? And the real problems with it are pneumonia and retinitis. It can be very dangerous to the newborn. But anytime you have a transplant patient and you transplant an organ, you have to test for CMV. And most of the time, you really don't have the time to do it. So they purge organs of lymphocytes and all sorts of blood cells in the hopes to get most of it out. And most of the time they do, and sometimes they don't. And if they have an opportunity to test for it, then fine. But if not, what you can do is introduce this virus into an individual who probably already has the virus. But the difference is when you do a transplant, you immunosuppress. And now you're going to reactivate that virus that's present. And that's more frequent than we'd like to know about. So how do you promote persistence? Doesn't seem like a really good thing for the body to do, but it does it anyway. Well, sometimes the innate immune system fails to clear an acute infection. And that's one way in which it can happen. Sometimes, because the viruses block apoptosis, which we talked about last week, that can lead to persistence. It allows the virus to become stable and associate and remain with the infected cell. What are the host contributions to persistence? Well, the two tissues that are void of initiators and effectors of the immune system are the eyes and the neurons. So while you have immune responses, the initiators really are, for the most part, not working up to snuff. And so they turn out to be a great place to start an infection because a vigorous immune response would be detrimental to the host. You'd be blind if you had a really good immune response to some of the viruses that infect your eye. And frequently people do get blind from them. So CMV retinitis or herpes simplex virus retinitis or adenovirus retinitis are all really dangerous diseases to those who get them. And most of them can be treated with antivirals. So these organs tend to be persistently infected as a result of the fact that the immune response is somewhat decreased. So how does the virus genome exist in these infected organelles? I've put up 3 states, and we'll talk about most of them. And one of them is a non-replicating genome in a non-dividing cell. So you all know that neurons don't divide. And both herpes simplex virus and varicella zoster virus are viruses that get into neurons. Somehow or other, they make copies of themselves, and it remains a mystery to most of us who have studied it. And they persist in the neurons at about 50 copies per cell. They persist as episomal units. That is, they're free of the host chromosome, and there's no evidence that they replicate. In other cases, we have autonomous self-replicating chromosomes in a dividing cell. Human papillomaviruses in basal cells of the developing epithelium, hepatitis C and B in liver cells, and Epstein-Barr virus in lymphocytes. On occasion, these viruses can integrate into the host chromosome and replicate with the host. HIV, parvoviruses, those single-stranded guys with the funny panhandle ends, and a human herpes virus called HHV6, which is a primary cause of a mild childhood disease. But an amazing thing happens with its genome, and we'll discuss that in a little bit of detail. So let's talk about some of the interactions that occur between viruses and their hosts. An interesting one is Sindbis virus, and there's a model in which you shove this virus into an adult mouse brain, and you can get a persistent non-cytopathic infection. So the virus survives that event, and so does the host. If you do the same thing into a neonatal mouse brain, you get a lethal infection. So in one case, there's a symbiotic infection, if you will. The virus survives, persists. In the other case, the host dies as a result of infection. Why? It's all about where you are and when. So in the neonate, neurons lack proteins that block virus-induced apoptosis. And when you apoptose, you die. Things necrose, you send out signals, you have a massive inflammatory response, and the animal succumbs to the infection. Another virus that provides an interesting paradigm is BDV. Anybody know what that stands for? Bovine something, right. Okay, so it's a bovine diarrheal virus, and it's a Pestivirus. And there are 2 strains of this virus. There's a cytopathic strain and a non-cytopathic strain. And it turns out that you can infect these animals in utero as a natural course of events, and the non-cytopathic virus will establish a lifelong infection in cattle. So it will persist in the infected animal when it's infected in utero. These animals have no detectable antibody or T-cell response to virus antigens. So they're making virus. The infected cells are elaborating new viral proteins, but they're not being seen. So what we say is the host is tolerized. It's tolerant of what's going on, and it just lets it go because it's going on at a slow, low level. When these animals are infected with the cytopathic strain, though, the interferon response is activated, and the virus is readily cleared. So 2 different strains getting 2 different responses. Dr. Racaniello talked about measles and SSPE last week, so I'll go through it rather quickly. As you know, there's no animal reservoir for measles. It's extraordinarily contagious, and if you isolate the infection, then you can stop the spread. There are 40 million cases a year, and it leads to systemic immunosuppression. So the sequelae of measles infection are more than just the overt development of complex spots and spots all over you. The good thing is that you develop lifelong immunity. So it's a great vaccine if that's the way you want to be vaccinated. The bad thing is that there are serious problems with those who are pregnant who acquire measles. And you saw this slide last week as well, where the normal infection leads to giant cells in infected tissues, spreads to the entire reticuloendothelial system, the virus gets shed, and then a secondary viremia occurs, and that's when you see the development of disease in spots. In rare instances, the virus gets to the brain, and there's an encephalitis that can develop. And even more rare instances, we see the development of SSPE, where only portions of the virus genome persist and virus proteins are made. And so what's the hypothesis about how this works? Well, in the infected individual, measles enters the brain in infected lymphocytes. Antibody blocks cell-cell fusion. So because the lymphocytes are in, you've opened up the blood-brain barrier. And the removal of fusion protein from the surface allows for persistence of portions of the virus. All hypothesis. None of this is fact. I mean, this is fact, but we don't know that that's how it happens. And then you get a slow infection that goes on that's not persistent. Low levels of envelope, no virions, but nucleoprotein complexes spread from cell to cell. It's a cytoplasmic virus. It's out there. It can go from cell to cell. And after a while, long-term accumulation of these nucleoprotein complexes leads to SSPE, and the individual dies in 6 to 8 years. I'm going to talk about herpes viruses because they manifest themselves in a large number of ways and elaborate sets of different RNAs and different proteins that are found under conditions of latency. So first we'll talk about herpes let's just talk about herpes simplex virus here. And I've told you before that an unusual series of transcripts originates from the genome in latently infected cells. And these transcripts are called latency-associated transcripts, or LATs, and they're derived from a single region of the chromosome. And they accumulate in the nucleus of the latently infected cell, and what they are is a stable intron. So normally they should be cut out of a messenger RNA, and they should be degraded in the nucleus, and they just accumulate, and nobody knows why. Its very close cousin, varicella zoster, does something entirely different. Its pathway of infection is similar. Its residency in the ganglia is similar. But it elaborates a small subset of proteins. And these proteins go to the wrong place. They're normally found in the nucleus. They're involved in control of virus replication, and they're found in the cytoplasm. Depending upon the type of EBV latent infection, certain virus proteins and small viral RNAs, like the adenovirus VA-RNA, EBV makes something called EBERS, E-B-E-R-S. And they're small RNAs that have highly structured molecules, and we believe that they act much like microRNAs. And they're required to maintain the latent state. If you knock them out, you don't get latency. And they're thought to modulate the host response. That is, whether or not the host sees the infected cell, how it sees this infected cell. EBV elaborates a IL-10 mimetic, and that has effects on how the host sees it. And then for HCMV and KSHV, not only did they tamper the host's immune response, and we talked about the CMV ways in ad nauseum last week, but they also elaborate microRNAs that are thought to play a role in the establishment of latency. Yes? Epstein-Barr virus encoded RNAs. And they're small, highly structured RNA molecules like the VA-RNAs of adenovirus. Okay, so how do you get CytoMegalovirus? I told you where it was found, and that should give you some clues. The virus can enter through the eyes, an ocular infection. It can enter through the oropharynx. It can enter through by sexual transmission. And it can be transmitted from a mother to a child through breast milk. So those are various ways in which the virus can be acquired. It infects epithelial and other cell types, including some of the lymphocytes. Most infections, though, are subclinical. You get the infection, there's no apparent display of illness, and you get over it. Cell-mediated immunity, however, is required for resolution of infection. And when the virus establishes itself as a latent genome in an infected cell, it does it in bone marrow progenitors and macrophages, so things that are going to be around for a long time in the host. And if you repress cell-mediated immunity, then what you get is recurrence. So once again, that relates to the topic of why you bother to deal with it in transplants and why it's so important. And all blood is typed for CMV among God knows how many other different viruses at this time. Infection in utero can be devastating to the fetus. Children infected in utero frequently are born deaf, have intelligence defects, have some anomalies, sometimes in limbs. Early childhood infection, less so. Once the fetus is formed, the virus has less effect. But the virus does persist, and it can be found in salivary glands and in nurseries where kids play with one another and smear themselves with God knows what secretions. They transmit it amongst themselves. And there's some great studies on this for those of you who want to look them up. And I've told you before, it's found in salivary glands, mammary glands, and semen. Reactivation can be with dire consequences. And the microRNAs that are expressed by CMV, both in vitro, when you take an organ out of an animal, and in vivo when seen in the animal, are tissue-specific. So certain microRNAs are made in certain organs, and that's very cool. So once again, pointing to the relationship between the virus and the host. And those microRNAs are associated with specific stages of virus infection in terms of virus surviving, virus being latent, virus being lytic, virus reactivating. All right, my first rule of latency. That which becomes latent must reactivate. Otherwise, there's no advantage, as the virus can no longer spread. So the fact that a virus comes in and latently infects you, it's not because that's really what it wants to do. That's a survival mechanism. It becomes a passenger for life that sometimes comes back to haunt you. Herpes simplex virus. The population in the United States is 80-something% seropositive for either type 1 or type 2. That means 250 million of us have latent virus hiding in our neural ganglia, and 40 million at any one time or in any one year will experience recurrence and some asymptomatic shedding. So when that cold sore comes, because it's finals time, it's because you are doing something to activate that virus. There are stress molecules that are known inducers of virus, among other things. How does the virus get to where it goes? And sometimes it can cause encephalitis. Sometimes and that's a very rare disease, but most of the time it just becomes resident in your neural ganglia. So the virus infects cells in the epidermis, either in the throat, on your actual skin, through a small cut, replicates in these epithelial cells and can spread from cell to cell. When it gets to the basolateral surface of the epidermis, it can then enter the dendrites, the long extensions of your sensory or your sympathetic neuron bodies. And then what the virus does is it goes up that very, very long axon, finds itself very happily ensconced in the neural body, and replicates to some extent. And that's the black box of this whole thing. And that's one of the things that we don't understand, and that is, why are there multiple virus genomes present in the nucleus of a cell that's not being killed, and really has no capacity for DNA replication because neurons don't replicate. Sometimes this virus can spread up to the central nervous system, and that's very, very rare. In the case of the sympathetic neuron, you've got to have contact with yet another neuron. But in each case, the establishment is the same. So what are the post-infection events in neurons? We know that the nuclear capsid, that structure inside the envelope, travels up the exon. But we also know that the nuclear capsid is stripped of all those ancillary proteins that surround it, the tegument. And so the viral activator of transcription, VP16, is separated from the nuclear capsid. So it's a little bit of a mystery as to how infection starts. There is limited productive infection. We don't see late proteins for the most part. You see it sometimes during the acute phase of ganglionic infection, but once it becomes latent, you no longer see those. And we believe that local inflammation, astrocytes coming in, lead to resolution of the infection. Genome is silenced, and it's coated by nucleosomes. So now it looks like normal DNA inside the cell. As I said before, there are multiple copies of virus DNA, anywhere between 5 and 50. And what's true for simplex is also true for varicella, at least for that. And then we see the nuclear accumulation of these latency-associated transcripts. If you look in a mouse model and you excise trigeminal ganglia at different times post-infection of the eye, and what they do is they scarify the eye and they put a little bit of virus in the eye of the mouse, and then you wait. And first the virus replicates locally in the eye, and then it gets transmitted through the trigeminal nerve to the trigeminal ganglia. And you see with time, the virus arrives, the titer increases, it replicates, and then by 2 weeks, it's totally disappeared. You can't identify infectious virus inside those ganglia. Now, when we talk about ganglia, we talk about thousands of neurons. And many of them are infected. And when we talk about reactivation, only 1 or 2 of those many actually reactivate. So the virus infection does not spread in the ganglia from cell to cell. What happens is the virus reactivates, it goes down that axon, and it erupts on the skin surface. And that's how you get a cold sore. All right, what do LATs do? So the answer is, I don't know, but this is what we do know. LAT minus viruses, so we can make those, we can remove the sequences that encode them, reactivate very poorly. There are 2 open reading frames that are contained in the LAT sequence, but no known protein has ever been associated with them. So there's no evidence that they get translated, or for that matter, that these become messenger RNAs and get into the cytoplasm. They do, however, encode microRNAs. And they encode microRNAs that could inhibit expression of ICP0, a potent transcriptional activator. That's the protein that takes apart the ND10's PML complex in the nucleus. And then another protein called gamma 34.5, which is a neurovirulence gene that activates, as you'll recall, protein phosphatase 1. So what it's doing is things that will inhibit the expression of these guys that will help establish virus infection. And when you look at the virus genome and you do computational analysis of the structure, what you see is that most of the sequences that could possibly encode microRNAs are found in the region of these LATs, or this overlapping gene ICP0 or 34.5. So these 2 genes are transcribed from one strand of DNA and the LAT complex from another. But in each case, we find multiple examples of microRNAs. And the other thing that we know is that while there are similarities and differences between HSV1 and HSV2, one is found predominantly above the waist, the other is found below the waist. Sometimes the one below the waist is above the waist. That could be a question for, how does that get there? That could be one of your final questions. All right, but you can figure that out. But what's important is that they're evolutionary conserved. So both viruses have this same set of microRNAs, and they're probably used to regulate expression of these gene products. Why neurons? Neurons are a great place to be if you're a virus, DNA virus that doesn't have to replicate. You've got multiple copies of yourself. You're primed to do something. You really don't know what, all right? But the cells themselves don't replicate or divide. The genome is readily established, and it persists. There's nothing there to take it apart. For the most part, it's insensitive to antivirals. So treatment with antivirals does not knock it down because antivirals generally are targeted to replicated molecules. No replication, no effective antivirals. They're insensitive to immune surveillance, and they're not violated for the most part because of the blood-brain barrier. But the question remains, how do they survive the primary infection? In any system in which we have looked, infection with a herpes virus of one type or another, where it replicates, kills the cells, except in the case where it establishes latency. And what's different about that? How does it get stopped? How does the apoptotic program get inhibited? And why do these things persist? So why are there multiple copies of virus DNA in cells that shouldn't support virus replication? I told you that only a small number of neurons in a ganglion reactivate. And that tells you that whatever the mechanism is that's responsible for reactivation and how those ganglia see or sense those signals, not all cells are equal. So some of them sense it, and they do something that allows the virus to reactivate. Virions appear in mucosal tissue that is innervated by latently infected ganglia, and blisters ensue. So you can think about it. I don't know how many of you have seen people with shingles, but shingles is the second coming of varicella. It ain't pretty. The really nice thing about it is that it's usually confined unilaterally, only one side of the body, and to a single dermatome. So a dermatome is an area that's innervated by a particular ganglion. And so that's why you see swaths of infected pustules along an arm or across a back or across the trunk of a body. Okay, I've told you that what happens to surrounding neurons is an enigma. The virus, which has all these beautiful little envelopes on it that allow it to pass from cell to cell, doesn't do that in neurons. So the adjacent neuron doesn't get infected unless the virus goes totally crazy. So there are evidences, for example, we've looked at hundreds of ganglia over the years where you can show that the virus reactivates in situ. And that's, of course, a devastating thing. But it usually, you have to remember that the tissues that we deal with are acquired at autopsy. And so it turns out that if you remove a ganglion from an individual and you wait a certain amount of time, the virus will actually reactivate from that axatomized ganglia, where it's not doing quite as well in the host. Many times reactivation is silent, but virus is shed. So that's a problem for infecting individuals. And there are enough studies on college campuses to demonstrate that that's true. How is the virus infection masked from the host immune response? Why is it that you don't, even though you have a great antibody titer, you have vigorous T-cell immunity, you still get recrudescence. You still have virus coming out. It may have something to do with the proximity of the dendrite to the epithelial surface. And that's conjecture, but it's the only thing that makes sense. Cells touch, and when cells touch and a virus comes down this long pathway, there's very little instance where the virus proteins are actually found on the surface of the axons. So the cell remains hidden from the host. What flips the switch? We know that stress is a great reactivator of herpes viruses. We know that glucocorticoids can reactivate. We know that sunlight and orange juice are effective mediators of those of you who are susceptible. And in a model system, we know that if we express this virus gene product, we can reactivate. But 2 years ago, there was a manuscript that was published using mice as a model. And this manuscript demonstrated that a virus protein, this VP16, the guy that is present in the virion particle, the guy that's responsible for initiating virus infection, and the protein that's made late in the virus infectious cycle, appears to be required and important for reactivating virus under some sort of a model system. And that represents a conundrum because it's thought to be a late promoter. But studies have demonstrated that funny things happen in neurons, and this gene is probably turned on early in response to exogenous forces. It's a tough experiment to do with people. You understand that, but we'd love to try. Okay, so compare establishment, maintenance, and reactivation. We have our primary infection with a virus, the introduction of its tegument proteins. You've heard all about this before. We get a class of virus gene products that are made immediately, and they modulate interferon and cytokine release, so they suppress the host a bit. Then we have early genes that are expressed, and we get DNA replication, and a commitment to productive growth, and the inhibition of apoptosis so that the virus can complete its replication cycle. Subsequently, late genes are expressed, particles assemble, and DNA is packaged, and in some instances, virus spreads from cell to cell. In some instances, it's spit out of the cell, and it goes into the bloodstream, and you have a viremia. Particles egress, infectious virus is produced, and then sometimes it goes into a different kind of a cell, and that's your neuron in this case. Immediate early gene expression is blocked, or we believe it to be blocked. Lat genes are expressed. Those RNAs accumulate. And latent infection is established. Viral gene products, not that we know about. The only viral gene products are, again, are that RNA and the accumulation of virus DNA. Virus is maintained in its episomal state outside the chromosome, God bless you, and certain stimuli that usually induce a stress response can lead to activation of virus replication. Latent infection, reactivation, you go back and you start lytic infection. Your pustule that results, your cold sore, results from lytic infection of epidermal cells and dermal cells beneath it to some extent. Now contrast this to some extent with chickenpox and shingles. Chickenpox virus, varicella zoster, is acquired either through the conjunctiva or through the respiratory tract. Replication is believed to occur in primary lymph nodes, so right up in the throat. After 4 to 6 days, sufficient amounts of viruses are made so that you have a primary viremia. So the virus is spreading throughout the body in its first course, and it lands in the liver, the spleen, and other target organs, where it replicates some more. And you have a secondary viremia. So it's not, one was simply not enough. And it's the second viremia where the virus spreads to the skin. And that's when you see the waves of development of chicken pox. How many of you have had chicken pox? Wow. Amazing. How many of you were vaccinated? 1, 2, 3. All right. So for the most part, people your age should have been vaccinated. Well, never mind. The vaccination is a difficult procedure. It's very difficult to get high-titer virus. And so the companies that make it, Merck and SmithKline, Glaxo, have had problems getting very high-titer vaccines. And as a result, kids get immunized twice with it. Now, or more recently, it's been shown that these vaccines are very efficacious to those of us who are slightly older and who may or may not have had chicken pox. You rarely get a second case of chicken pox, but you do get shingles. As you advance past the age of 55, the occurrence of shingles increases. And we believe that's because the T memory cells go to hell after about 50 years. So you lose your immune response. So people get boosted, and there's now a vaccine for that. Anyways, we have this secondary viremia. You get infection of the epidermal cells, and you get the characteristic rash and little pox, and they then spread to the sensory neurons. There's no evidence that the sympathetic neurons are infected, though I find it hard to believe that they escape. Again, that's my prejudice. Anyways, they go and do what simplex does, and they sit in the ganglia. So these are immunohistochemistry pictures of human ganglia that have been sectioned and surveyed for the presence of varicella zoster virus proteins. And we note that in the skin, when you look for VZV proteins, this protein in particular, ORF29P, it doesn't matter what it is found in the nucleus. And it's a protein that's required for DNA replication. So here's ORF29 in a neuron body. And you'll note that the nucleus has nothing in it. All of the protein that is present in this neuronal body is present in the cytoplasm, as is a protein called ORF21, 63, 62, and 4. And all of these are regulatory proteins that the virus elicits during its normal course of productive infection. The difference is those proteins are found in the nucleus where the virus does its dirty work, and they're not found in the nucleus in neurons. So there are 5 of these virus-specified proteins. Each is a regulatory protein that controls either gene expression or DNA replication, and they're aberrantly localized. Now, Epstein-Barr virus is a gamma herpes virus. "95% of adults are seropositive and carry the genome." How many of you have had mononucleosis? You can't say you've been to college unless you've had mono. All right? The virus resides in persistently infected, non-proliferating B lymphocytes, and it's the causal agent of all of these different cancers. And it does that because it goes latent, and it changes the cell programs and causes these cells to replicate under unusual conditions. The EBV lifestyle is, and this is just one example of it, so don't commit this whole thing to memory because there's more to it than that. What I'd like you to recognize is that in a primary infection, and we're starting up here in saliva because that's the normal portal of entry, and mononucleosis for years was called kissing disease because that was how it was most evidently transmitted. So you get infection of epithelial cells where the virus is thought to replicate poorly but get to resting B cells. And it's there where the virus really finds its great happiness. It goes in, it synthesizes a few proteins, and it can either replicate, and if it elaborates both a latent membrane protein 1 and a latent membrane protein 2 and other EBV proteins, then CTLs will see it, killer cells will see it, and the infected cell will be killed, and maybe virus will be released. And if virus is released, you can go back and infect more B cells. And then you sometimes see a different kind of an infection where only this protein, EBNA1, Epstein-Barr virus nuclear antigen, and LMP2 are found. LMP2 is a stealth protein. So it's found along the surface of a cell in the outer membrane, but so little of the protein is exposed that it's not recognized by CTLs. And these cells can live very happily with Epstein-Barr virus, very quietly, and once in a while they get activated to make virus, and the virus comes back out through the oropharynx and contaminates your saliva and the next person you kiss. In latently infected B cells, the virus chromosome is a self-replicating episome. It associates with nucleosomes. It's methylated like normal cell DNA is at CpG residues, so it looks a lot like the host. For the most part, it expresses LMP2 and one of those EBNA guys, and these cells home to bone marrow and lymphoid organs and rest there. They're not seen by CTLs or virus-specific antibody because the only thing on their surface is this stealth protein, LMP2. But virions are produced in a very small fraction of the cells. Those are the guys that get reactivated, and so they get spread. I told you before there are multiple forms of EBV latency. And it turns out that they do different things to the host. In some case, they produce EBNA1 and those small EBRs, those small RNAs that are actually Pol3 transcripts. Sometimes they produce these guys and LMP1 and LMP2 and other microRNAs. And sometimes you have a third type where they do all of the above, plus a protein called EBNA2, EBNA-LP, and these activate host cyclins. And once you activate a host cyclin, you turn the cell on to replication, and you promote cell division. So one of the ways in which monoclonal human antibodies were first made was to activate B cells, was to infect B cells that were plasma cells with EBV and select for those that proliferated, and they would overexpress the antibody that they were synthesizing. So what happens when a B cell divides? The episomal virus genome has to replicate to be distributed to daughter cells, and it has to replicate in synchrony with the host. Again, if the host divides, the virus really wants to be in both of those cells, both the parental cell and the daughter cell. And it's evolved a very, very interesting way of using the host to control that. It has two origins for DNA replication. One, or ELIT, is used for lytic replication. That's when the virus needs to make high copy numbers of virus, lots of genomes. The other, or EP, is used for episomal replication in latently infected cells, low copy numbers of virus genomes. So replication of this whole thing, as I told you, has to be synchronized with the host. We know why. It's to preserve its genome in the different cells. OREP is normally quiescent. So this origin that's used to replicate in latently infected cells is not used during lytic replication. And the reason that it's quiescent is it's bound by host regulatory proteins that block EBV DNA replication. And EBV elaborates a protein called EBNA1, which is a nuclear antigen, which binds DNA. And once bound to DNA, it interacts with host proteins to form a stable complex, an origin recognition complex, or an ORC. This replication that occurs in latently infected cells is in synchrony with a host. So you remember M, S, G1, G2, all those guys that are extremely pertinent to how EBV replicates. So replication is said to be licensed by the formation of this origin replication complex. And what happens is host regulators are released and virus DNA initiation occurs. The host proteins that are involved in controlling both initiation and repression are synthesized and degraded as a result of the host cell going through the normal replication cycle and cell division cycle. And this is a picture of what goes on and what we refer to as replication licensing. In M, in mitosis, the Epstein-Barr virus genome is bound by this origin replication complex. During the course of M, there's a protein called geminin, which binds another host protein. These are all host proteins now, CDT1. And as M progresses, geminin is degraded, CDT1 is released, and one of the cell cycle division proteins, CDC6, which is a protein kinase, is also synthesized. In G1, these are brought to the origin replication complex, and they form a more complex complex, if you will, and they act as an attractant for another set of host proteins called MCM. MCM binds to the DNA, brought to it by this complex of proteins between the ORC and CDT1 and CDC6, and forms a pre-replication complex. Once this complex forms during G1, the cell enters S. And S signals CDC6 to phosphorylate proteins on the origin replication complex, and geminin is released with CDT1. You now have what, for all essential purposes, looks like your SV40 replication origin. So you have bidirectional replication of this very large 190 kilobase genome and release of CDC6, which in G2 is degraded, release of MCM, which enters the cytoplasm and gets phosphorylated and inactivated, and geminin taking CDT1 out. As the cell goes back into M, we see that CDT1 is released from geminin, it gets degraded, and the whole process starts over again. So that's replication licensing, allowing the virus genome to replicate coordinately and concurrently with host chromosomes. HHV6 is a beta herpes virus, and it's a causal agent of a very mild childhood disease called exanthem subitum. And everybody's got it. Everybody's seropositive for it. The virus has been found to persist in T cells, and it infects the host for life. But unlike the other herpes viruses that we've talked about, EBV, CMV, VZV, HSV, there are no circular episomal forms. In fact, what's found is that the virus genome integrates into telomeres. So those very ends of the chromosomes that have duplicated regions of DNA. And the reason for that is that it has sequences that are very similar to it, and it's a very easy pathway for it to get in. And it can also get out, so it can be reactivated in response to certain molecules, and probably also in response to immune suppression. So this strategy of lodging yourself at the ends of virus chromosomes makes integration a plausible molecular strategy for viral latency. And in fact, it's one that's in part used by some of the Papillomaviruses. Now, there are over 100 distinct types of human papillomaviruses. We now have a vaccine that protects against most of the types that are involved in formation of cervical and uterine cancer. And that's good. They don't protect against everything. But these viruses segregate into what we call mucocutaneous and cutaneous types. That is, they infect mucocutaneous areas, the genital areas, or regular skin, dry and wet surfaces. And in each instance, there are both high and low risk types. And what I mean by that is low risk types form warts, benign papillomas. High risk types have a predilection towards causing cells to be driven into the cancer phenotype. That doesn't mean that they survive or that all of them go on to form malignant infections, but there is some incidence. When a papillomavirus enters the skin, either the cutaneous or mucocutaneous surface, it infects the basal layer. So all the way down, right where the epithelium starts, and right at the surface, the junction of the dermis. And it first replicates as episomes as cells divide. Remember that your skin, being the largest organ in your body, also turns itself over quite frequently. And that's a result of these basal cells dividing. And they replicate as theta forms. So instead of coming out in those big molecules, the way we talked about SV40 and Papillomaviruses, they don't replicate bidirectionally. It's just in one direction, so it's unidirectional. The viruses replicate their genomes and make proteins only in terminally differentiated epithelial cells. And these are cells that get sloughed off. When they're going to become, let's see, how am I going to put this? When they're not replicating, that is, when they get into cells and they get integrated into the virus, into the host chromosome, and you see changes, dysplasias in cells, that is, they don't look normal. They have the capacity to divide in places where they don't normally do. So it's not just the basal cells anymore that are dividing. It's those in the stratified epithelium. What happens is the viral chromosome is into the host, and that interrupts the program of terminal differentiation of the host because of the expression of these two proteins. And these two proteins act to interfere with cell cycle regulators. So they push these cells from being limited in terms of replication and being focused on differentiating into being replicating cells. So this is what it looks like. Here's your skin. And here's that basal lamina and the precursor skin cells. These are pluripotent stem cells. so that they have the capacity to become many other tissue types, but more importantly, they're self-renewing. So each time they divide, they give rise to a mother and a daughter. And each time they divide, they push the layer that's above them up further. And as these cells get pushed up into the further and further into the epidermis, they begin to differentiate. They flatten, their nuclei begin to decrease in size, and a whole different program of cell surface proteins that lead to the elaboration of keratin is initiated. In these more differentiated skin cells is where the virus normally replicates when it's undergoing lytic infection. In the case where the virus actually gets integrated, then these cells acquire the capacity to replicate by themselves. So they're no longer terminally differentiated, and they can spread and cause a lesion. Papillomavirus persistence. Intact virus genomes persist in basal cells of developing epithelium. The genomes divide as episomes with a host. Again, just like Epstein-Barr virus. Each time that cell gives rise to a daughter cell, that virus genome gets passed on. Infectious virus is not present except in the stratified part of the epithelium. In developing cancers, the genome is integrated. It replicates only when the host cell divides. Again, it's part and parcel of the host chromosome. And virus proteins such as E6, E7, and a membrane protein, E5, are expressed. There are a family of viruses known as the human Papillomaviridae, and there are now 6 known members of this, and they have these great names. The V always stands for virus, by the way, and the other things stand for something, probably where they're discovered or I don't know what. But most of these can actually cause tumors in animal models. Does that mean they cause tumors in man? Only Merkel cell virus is associated with a human tumor. The other human Papillomaviruses appear to latently infect people. And for the most part, that's rather benign. But, what did I do? No, that's no good. Okay. It turns out that people who have JC or BK are frequently found to go, it's not frequent, are found sometimes to go on to develop a disease called progressive multifocal leukoencephalopathy. Remember, that was what you got to say this on the way out for your final. And under these conditions, myelin is lost and not replaced by oligodendrocytes. The nerves become damaged, and everything stops working properly. And we've seen a rather higher incidence of it lately because people with multiple sclerosis have been treated with an immunosuppressive antibody called Tisabri. And so this is a balancing act. We know that most people have these viruses in them. And we know that in most instances, treatment with this antibody does not result in reactivation of viruses. But in a finite number of cases, it does. And they develop PML. So that's a sad state. It's going to get them, either the disease is going to get them or PML is. But the bottom line is Tisabri is a very effective drug when it's working properly. So a famous guy in a blog in 2009 said, given the high seroprevalence of papillomaviruses in humans, it is not surprising that they are significant pathogens in immunosuppressed populations. An important question is why these viruses can peacefully coexist in many humans without causing disease. Are human papillomaviruses simply passengers, or do they benefit us in some unknown way? And that can be said for almost any virus in which we talk about latency. So let me finish with something that was found a couple of years ago. This Merkel cell carcinoma was actually found and discovered by the same people who discovered Kaposi's sarcoma herpesvirus. And it turns out for all the world, and the reason that you can't read it is because it's not important that you read it. Just remember that it's a circular molecule, and what I want you to know is that it makes a large T antigen, a molecule that's homologous, orthologous in this case, to the SV40 T antigen, that molecule that does everything. It does replication, it does transcription, does cell transformation. And when you look at it on a phylogenetic tree, you see that it's really not that close to the other human Papillomaviruses. Rather, it's a little bit closer and off of a tree in which African green monkey, murine, and hamster came. And that tells you absolutely nothing. What is interesting about this virus, though, is that in tumors, when the virus is present, and Merkel cell is a very rare tumor of neuroectodermal origin, and it evolves from the Merkel cell, which is a mechanosensory receptor. And what we see is that virus DNA is integrated in a clonal pattern. That means one cell got infected and expanded. So the integration site is some novel place in the cell genome. So infection and integration preceded clonal expansion of the tumor cell. The other thing that's interesting is that all MCV-positive tumors have mutations in T antigen. So they don't have fully proficient T antigen molecules, and therefore they're replication deficient. So when the mutation occurred is not clear, but we know that the integrated virus genomes cannot be excised and that the cells survive because the virus doesn't replicate. So let me finish with this thought about persistence, and that is that viruses preferentially target slowly dividing or non-dividing cells to host their latent genomes because it's the best place to hide. If a cell ain't doing anything, it's going to be left alone for the most part. They adopt a variety of survival strategies that coordinate replication of their genomes and expression from these genomes to allow them to persist. And in response to a variety of stimuli, these latent genomes can, on occasion, reactivate. Thank you.